Good afternoon, class. So, in today's botany class, we are going to see the how we can see the seed structure and their dispersal mechanism. So, very first, we have to see very interesting topic, the seeds. You can say that seeds are the basic structural and functional unit of the plant. Like if the seed is not going to germinate, how a plant will be formed. So basically, seeds are a very important unit inside the plant, or they also very you can say determined for the life cycle process of a plant, because if there will be no seed, then there will be no new plants to be formed. So that's why seeds are very important. We have to see, like if we wanted to see the introductory part of you can see the definition of seed. So basically seed is a kind of tissue inside a plant which have a generative values, or you can say they have that span which will be converted into a whole plant. So, or in some another word, you can say that they are possess inside a very little embryonic plant inside them. So, when they get all the favorable condition, they are going to convert that embryonic plant into a full-fledged big tree. So, if we see that where these seeds are present, so basically, plant has been categorized under two broad categories. One is a phanerogams, and second one is a cryptogam. So the phanerogams, which are having the seed in them, so you can also call them the seed plants, phanerogams. And another hand, there are the cryptogams, basically the plants which do not have seed inside them. So this kind of plant, like the cryptogams, in which we are going to include the bryophytes, thallophytes, and pteridophytes. So all these three divisions, they do not have seed formation inside them. So you can see the seed is a very fundamental or sometimes a characteristic feature of the phanerogams inside the plant classification. If we wanted to see how a seed structure will be look like in an overview, so they're having this firstly a very coat, or you can say the seed coat, which is the outer covering of a seed, which helps seed to prevent from harsh condition, environmental stress, and many other toxicological environment. And inside the seed, they are having the stored material, embryonic plant, which are very important reproductive structures inside the phanerogams. So, if we talk about the structure of seed, that this can be very easily studied by very common type of the seed like pea, gram, beans, almond, sunflower. So there are abundance of the seed by which we can study the how seed going to be germinate, what are the features present inside the seed, and how they are different from other part of the plants. See, though all are built on the same plane, through there may be differences in the shape, size of the seed, and the relative proportion of various plants. So, the seed, the propagation of the seed, they have the same kind of process, okay? Like the germination of the seed, they do not have a very differentiation among them. But if we are talking about the shape, size, and structure, they can be vary from species to species. There are hundred of variations in the seed size, shape, color, and surface. So you can see that there are a lot of kind of the size which can be present, shape, different shape, color. Like most of the seeds have some dull brownish color, but they can be very. Some seed have bright red also, yellow also, white also, but most of the seed you have been seen that they are brownish or dull in colors. Surface also, they have a very different kind of surface. Some have a smooth, some have some wrinkled, some have lines on their surface. So this can be a very kind of surfaces can be present. The seed ranges in the size from tiny dust particle as found in some orchid. So if we see the variation in their size, so it can be from the very tiny dust particle like seed inside the orchid and to large double coconut. So you all know has been how the coconut plant seeds has so big because you ate coconut, so you can see the that hold the coconut you ate, that is a seed of coconut. So you can see how large it is. And if you are going to see the seeds of orchids, they are very tiny. Even sometime you cannot identify them by your naked eyes. They are like dust particles. So the seed surfaces, they can be smooth, wrinkled, striated, ribbon, furrowed, reticulate, tuberculate, alveolate, hairy, pulpy, and having some sometimes finger-like prints on them. So basically, there are a lot of variety under the seed surfaces. Smooth, as you all know, they have no scars on them, just a plain, flat, fleshy. Smooth, wrinkled, maybe some wrinkles will be present on them. Straight, so they can be straight linings. Ribbed, some rivet version can be present. Furrow, some depression can be happen. Reticulate, the reticulate thickening, if you have seen in the tissue when I taught you the plant tissue system. So I taught you about the furrowed and reticulate thickening, tuberculate thickening. So these type of, you can say, these are the line patterns which can be present on the seeds. I can show you the diagram here. This one you can see they're very smooth. Okay, nothing, no scars will be present. Okay, so this is very smooth. They are this. They sometimes they are this furrow and depression inside them. This is straight. The lining, parallel lining can be present. This is that they are having some some rib-like structure. Reticulate thickening, you can say this is a reticulate thickening can be present on them. Okay, so these kind of variations can be present on the surface of the seed. So that can be very different or vary from species to species. So you cannot say that very specific terms, or you can specific features about the shape, size, and color of seed. So that can vary from species to species. So also we can see that these have some very differences in the shape and size. You can see there are a lot of kind of seeds are present here. Every seed have a different color, and their sizes are different, their textures are different, and they are having a numerical or range of the size. So this cannot be a definite to anything, like every species have their own characterized shape and a special kind of seeds present inside them. Now, in the seed, life activities are temporarily suspended in order to enable the part to successfully pass through unfavorable and injurious climatic condition. So, when the seed in the seed stage, okay, or you can say when the plant in the seed stage, so all the life activities like reproduction, like respiration, like gaseous exchange, transpiration, and every activities which can be performed inside the plant, that can be temporarily suspended. So they are just waiting for the favorable condition, and under the unfavorable condition they are not going to perform any kind of lifeful activities. So they are just waited for the favorable condition. When they are going to get the favorable condition, they start developing or they start going to germinate. On the approach of the favorable condition, the seed resume activities, active life, grow into full plant in the form of the seed, and a plant can be carried to long distance without special precautions. So basically, whenever they are going to have some favorable condition, like if we are talking about the favorable condition, so what kind of condition we can talk about? Like they are having a proper water facility, they are having a nice climate, or you can see the favorable climate for their growth. Some seed can be grown in the summer, some seed can grow only in winter. So basically, the suitable conditions in which a seed can be grow, like a suitable condition in the sense of light, temperature, and water content, humidity. So these are the environmental factors that can be play a major role in the growth and development of seed. So if the all the things are according to the seed, or you can say, or the flexible for the seed development, at that time they are called as a favorable condition. So seed just have to stop their activities till the time they are going to get all the favorable condition inside them. So in the form of seed, a plant can be carried a long distance without special precaution. So when a plant is in form of the seed, they do not need a variety of precautions. They just can be carried it throughout their life, or you can say a very long time distance can be carried inside the seeds. Now this is all. This was all about the important feature of the seed. Now what we have to see, the second thing, that is a structure of seed. How the seed look alike. So the various part of the seed may be easily studied after it has been soaked in water for a day or sparing according to the nature of the seed. So basically, if you wanted to study a seed, so we needed it to be a little soak out, because when we get it to be soaked, then they just swell up, and when they just swell up, we can easily study the all the membrane or the all the part of the seed, because when they got to increase in their surface area, when a thing is little bigger in size and you can say they are freely open, then only we can easily identify all the characteristic feature from the inside. So that's why the soaking of the seed for a day or two, that depend on the nature of the seed, how easily they get, you can say they absorb moisture. So on the basis of that, we can study the seed anatomical portion. A mature seed contain an embryonic plant, as we already studied that inside a seed there is an embryonic plant. So, what is the embryonic plant? What at what condition we are calling it embryonic plant? So, this embryonic plant is nothing, just a combination of radicle and plumule. So radicle and plumule, that are the major parts which are going to convert into the root and shoot portion of the plant. So this embryonic plant, that's why we term that the embryonic plant, because at this time they are not fully flesh developed, but they are a prototype of the root formation and shoot formation. Radicle, that will be going to convert into the root, and the plumule, that will going to convert in the shoot, is provided with reserve food material and protective seed coat. And with this radicle and plumule, they are having the reserve food material. When the in last lecture I told you about the endosperm formation. So endosperm formed there just to provide nutrition, or you can say for the nourishment of the seed. So in the reserve food material, or you can say that is present in the term of the endosperm. So endosperm is present there, so they can nourish the developing seed. And the protective seed coats. Last and very important, that is a protective seed coat, which basically give protection to the all the inner things like embryonic plant as a food material. So if they are now seed coat or seed covering, then there will be exposure to the environment, and these internal things, which is very soft, they can be harmed or destroyed. So that's why a proper coating of the seed coat and seed cover, that has to be present on the outer side of seed. A mature pod of the pea, Passum sativum, pea that is very famous for every study from Mendel to today. So has a number of seed arranged into rows. So we already seen that the mature pod have a number of seeds inside them. So that pod, that covering, the green cover, that is a seed cover in that case. This is a structure, or you can say the developmental phases of the seed. The very small seed is starting developing and getting out the embryonic plant from inside to the outside. This, if they are having this one cotyledon feature, this will be called as a monocotyledon plant. But I think they are having this two leafy appearance on their top, so that's why we can call them the dicotyledons. We will take it later. So the seeds are attached to the fruit ball by a small stalk and the funiculus. So the very first thing here, the attachment of the seed. So seeds will be attached to the fruit. Okay, seed have to be attached to inside, present inside the fruit. So very initially they have to be attached by a small stalk, that is called the funiculus. funiculus. Okay, we already studied about the funiculus in the ovule structure. So at maturity, on one side of the seed coat, a narrow elongated scar representing the point of attachment of seed to its stalk is distinctly seen, that is the hilum. First, very important thing, that is funiculus. So basically funiculus help the seed to attach with the fruit wall. Now the second thing is helium, which are going to help to attach the seed to the funiculus. Okay, funiculus will be attaching to the seed and funiculus also attaching to the seed fruit wall. So that attachment portion, at what condition the funiculus have to attach with seed, and that attachment point on the seed is known as a helium. Now, close to the helium, situated at one end of its there is a minute pore that is called micropyle. So, with the very close to the helium, there will be a end or there will be a pore or minute pore that is called as a micropyle. So, we already studied about the micropyle in the ovule structure, that this is the end where where the embryo will be formed, or you can say where the pollen tube get present or releasing the male gametes. So during the seed germination, water is absorbed mainly through the spore and the radicle come out through it. So this micropole, or you can say this micropyle pore, which has to be present near the helium, that have a very important role to play under the seed germination. How it is going to be useful? That the water which we give the water to the seed to germinate, so the whole absorption of the water is going through happen this pore or this pole, and when the root will be start becoming, or you can say the root is starting to germinate, then time radical have to be come out from this micropyle and goes into the soil. Continues with the helium, there is a short of rise in the seed coat, the raphe, and the seed is covered by two distinct seed coats. Now, after that, helium, there also have a ridge in the seed coat, or you can say some kind of depression that will be present in the seed coat, that is called the raphe, and the seed covered by two distinct seed coats. Now, seed have to be covered by the seed coat. So there are two distinct seed coat. The outer whitish one, or you can say the outer hard whitish one, that is called the testa. Okay, outer covering is known as the testa, while the another inner thin hyaline and membranous covering, that is called the tegmen. So basically, inner lining of the seed coat, this will be soft and membranous, and a hyaline, that will be known as tegmen, and outer layer, that is the wheatish one, very hard core, that will be known as a testa. So the seed coats give necessary protection to the embryo which lies within. So basically, what is the main function of the seed coat here? That they are going to give protection to the embryo while they are developing. The wheatish fleshy body as seen after removing the seed coat is the embryo. So after when we are going to peel off that wheatish fleshy body, so there will be a thing which we are going to see, that is the embryo. So now if we talk about the embryo, if it consists of two fleshy cotyledon and a short axis to which the cotyledon remain attached, so they can be two fleshy cotyledon and a short axis. So this can be structure we can see, and the position of axis lying outside the cotyledon, bent inward and directed towards the micropyle. So now what will be the position? The bent position of the axis, that will be bent through the micropyle. Because whenever the germination will be going to happen, the radicle will be come out from the micropyle end. That's why this depression is towards the micropyle pore or micropyle end. So the micropyle is a radicle, and the other portion of the axis lying in between the two cotyledon is the pleuron. Now the another axis which is opposite to the radicle in presence, that will be called as a pleural. Now this plumule in this one, okay, this is always the outer one, that is will be known as a plumule, and the inner one going inwards in condition, that will always be known as a radicle. So radicle is going to make the root portion, while the plumule that is going to make the shoot portion of the plant. So the plumule is crowned by some minute young leaves. You can see that, okay? Plumule, they are just minuted by very beautiful small leaves. The radicle give rise to the root, and the plumule to the shoot, and the cotyledon store of food material. So all these three things which are present after we peel off that British layer, they all have specific functions to do inside a plant development. Plumule will be going to the shoot, radicle will be going to the root, and the cotyledon which present along them, they will be work as a reserve food material. Since the reserve food material is stored in a massive cotyledon and the seed lacks the special nutritive tissue, the endosperm. So now, if the cotyledon is here, so there will be no endosperm present, because endosperm just till the zygote formation and giving embryo to their nourishment. But now they are converting into the seed. So when they are having the seed at the maturity, so, or sometime if there, sometime the endosperm will be present at the seed maturation, but sometime the endosperm has not been present at the seed maturation. So, on the basis of that, also we classify the seeds, like the seeds which lack endosperm at maturity, they are called as non-endospermous or Eczelaminous seeds. So, which you can easily remember that by non-endospermous seed, which do not have endosperm at the time of maturity. On the other hand, in several other plants such as castor beans, coconut, some cereal, food is stored in the endosperm. So some plants they are having this food storage in the terms of the endosperm. So that such seed where endosperm persists and nourish the seedling during the initial stage, they are called the endospermous or albuminous seed. So, basis of these, like presence of endosperm during the maturity or not, can also be a basis of classification of seed. If they are present, they will be called as endospermous seed. If they are not present, they will be called as non-endospermous seeds. This is the diagrammatic representation of a seed. You can see all the character which we studied here. This is the raphe. This is a challenger point. This point is called as a micropyle point. This is helium from which seed is attached to the funiculus. Micropyle and the position at which you can see radicle will be come out and form the root. This outer covering, that is the testa, and this inner covering, inner layer of the seed, that is a tagma. So outer hard layer, that is the testa, and inner fleshy membranous structure, that will be known as tegmen. And these do broad things, do broad leafy things look like they are the cotyledon, which are having the reserve food material for the development of plumule and pedicle. This is a plumule, a little above to the axis, or sometimes we can call them the epicotyle because they are present on above to the cotyle. So that's why we can call them the epicotyle. Sometimes we can call them the hypocotyle because they present downwards to the cotyledon. So that's why epicotyle and hypocotyle ultimately they have to be form. Epicotyle have to be converted into the shoot, and hypocotyle have to be converted into the fruit. So this is a basic structural analysis that has been present inside the seed. Now, on the basis of the number of cotyledon in the embryo, the angiosperm have been divided into two large groups. So basically, the number of cotyledon, these angiosperm divided into two largest group. The one is dicotyledon and second one is a monocotyledon. As their name suggesting, di means two. If the embryo having two cotyledon, they will be called as dicotyledon. If the embryo having only just one cotyledon, they will be known as a monocotyledon. Now, if we talk about a maize grain, that is a single seeded fruit in which the seed coat, fruit wall are unseparable. So in maize grain, basically, this is a single seeded fruit, no double thing here, no separation inside fruit wall. So on one side of the grain, a small, opaque, whitish deltoid area is seen to be distinctly marked out from the region, and the embryo hairs lies embedded in this area. So there is only one thin layer surrounding the whole grain. So sometimes this can be happen. So this whole layer things which have to be covered, this is to be very distinctive. But in the maize grain, which have been we taken an example for study, they do not have the distinctive layers between them. So now what happened? This layer is made up of the seed coat and the wall of fruit fused together. So the grain remain divided into two unequal proportion by a definite layer known as the epithelium. So the bigger portion is the endosperm and the smaller portion is the embryo. So the endosperm is a food storage tissue and the embryo consists of the one shield-shaped cotyledon known as scletum or axis. So here in the case of maize there will be only one cotyledon which have to be present. So sometime when the one cotyledon has to be present inside a plant, there have to be an endosperm which going to help the plant or seed to get mature by giving them the nutrition in a maximum amount as they need it for their growth and development. Now the upper portion of the axis with minute leaf arcing over it, it is plumule, and the lower portion provide with the root cap and the radicle. So we already know that the upper liable portion of the cotyledon that will always be considered as a plumule, which going to be converted into the shoot, and the radicle that will going to have a root cap and the root inside them. This plumule is surrounded by the leaf sheath or coleoptile and the radicle is surrounded by the root sheath or coleorhiza. So there are two new terms here for the one which is going to cover the plumule, that we can call them the leaf sheath on coleoptile. Okay, and second one which is going to cover the root hair, that will be called as coleorhiza or root sheath. So that just the name given after their features, like the someone covering the shoot leaves, so we call them the leaf sheath. If someone covering the root one, so we will call them the root sheath. So these are the protective sheets of the plumule and the radicle respectively. So basically these which sheets which we are talking about here, they are the protective in nature or they present there just for the protection of both plumule and radicle. This is a structure of a seed. You can see them here. This is the root cap. This is coleorhiza, which I just told you that the sheath which covered the root cap or radicle. And this is the coleoptile, which is a sheathing leaf which going to cover the lumen. So this is near the downward condition, they are having this radicle. If above one condition, they are having this plumule, this is going to convert into the shoot, and this needed to be protected till they are going to convert it into the shoot. This is a remains of style and this is a fused pericarp and testa. Their testa and pericarp, they got fused when they are developing. So this is a normal mesh grain structure. Now, besides the basic structure like the endosperm, embryo, and seed coat, there are some special structure may arise during the seed development. So these were the very common structure and going to be present in each and every kind of seed like endosperm, embryo, and seed coat. This is a very basic type of features. But in castor bean, a fleshy whitish tissue that is called craniole develop at one end of the seed and it is derived from the integument. So this is a new feature, that this feature craniole that basically develop from the integument. Integuments are the covering in the ovule. So if a new feature can be happens, so we can call them and give them a new name. The juicy edible part of the lychee fruit that is called aryle is an outgrowth of the funiculus that development after fertilization. So you all see the lychee fruit, and that white juicy part, that is not a part of a seed at all. That is a part of developed funiculus that develop after the fertilization. So this whole thing which we are eat in the lychee, that is the funiculus only, not even the pericarp or mesocarp or fruit. That is a part of ovule which develop inside the pulpy zone. The cotton fiber, they are the elongated epidermal cells of the seed coat. So this is also interesting that the seed coat, they are having them some internal, or you can say some epidermal cells of the seed coat, which going to be converted into these long hair of cotton. So that's why they are present. Mostly their function is for the hair, for the protection, and also after when they the seed matured and these fibers grow, they will be used as a commercial cotton, and we can make clothes from them, and that will have a very huge range of their functions. So now this was all about what is a seed. Now what we have to see, that is a seed dispersal. Seed have to be dispersed from one place to another place, because if seed not going to be dispersed from one place to another place, then there will be no mutations, no generation of new plant at other locations, pollination. So these have to be done by the seed dispersal process. So this is very important process here. So this is an adaptive mechanism in all seed bearing plants. So this seed dispersal mechanism that happens in all the plants, and they have to be this process, or you can say participating in the movement or transport of seed away from their parents plant to secure the germination and survival of some of the seeds to other adult plant. So basically in this, the seed have to be travel far from their parents plant and get to germinate some other plants. So in that way they have to be transported, and vegetation got the diversity. If the all the plants which present there, they cannot just grow in the shadow of their own plants, they have to be present somewhere else to grow nicely. So that's why the seed dispersion mechanism is very important. Now, what we have to see the type of the seed dispersals. So, the very first are important. There are lots of type, like 4 to 5 types by which seed can be dispersed. Very first, what we are going to see here, that is the seed dispersal by wind. The wind, this is natural and fundamental, means the seed dispersed in the plant kingdom mostly by the wind. When the wind flows, the seed, which are not very tightly attached to their parent plant, they can be just flow out by the wind pressure and go someplace another. So this process of dispersal is mainly seen in those plants which bear light seeds. Okay, because wind cannot carry a very hard thing. So the light-weighted seeds that can be only transferred by the seed dispersal by seeds. So seeds of the orchid plant, dandelions, swan plants, cottonwood tree, hornbeams, ash, kettle, pula, willow. So there are lots of light seeds bearing, so they can all be transferred by the wind. Now the seed disposal by the water. So water also have a very high flow rate. So when the rain come or we give the water to the plant, at that case also they can be go away from their parent plant and germinate. And sometimes like the lakes, ponds, some fishes also can let them from one place to another place, or some big animals having on their feet, animals of the pond or lake only. So that's why they can be dispersed by the water also. So there are some very important kind of plants which can do their seed dispersal by the water, like the coconut, palm, mangroves, water lily, water mint. So basically these mostly hydrophytes plant. They are responsible by the dispersal of seed by water, because as they lived inside the water. Now, the third and very important seed dispersal by the animal and the birds. So they are the most fond for the having the seeds to eat fruit. So that's why they just go to the different plant, eat the fruit material, and just take out and the seed and leave it in another ground, and that will going to help the seed to be germinate. So now few animals and birds, they are attracted to the bright colorful fruits. They eat the entire fruit and only the juicy part is digested by their system, and the seed are excreted out in form of their drooping, so which form into a new plants like blackberry, cherry, tomato, apple seeds and dispersion in this way. So basically, what sometimes they just eat up all the fruit, but they cannot swim. The seed particle cannot be digested by their system, so they just poop it out, and wherever they just excrete it out, the new plant get germinated. Some species like the squirrel, they collect nuts from the different plants like acorn and bury them under the soil. They literally doing the plant germination process. They're doing that for the storing their winter session, storing the food for their winter session. But after some time, what they do, they just forgot where they hidden their nuts. So after some time, the nuts which they have been buried, they will be grow into a full plants. Dispersal by gravity, as the gravity is a force of attraction and that will be apply on all the objects in the universe. So here also, when the fruit falls, that will also fall by the force of the gravity. So sometimes what happen when the fruit wall is not as heavy, and when they drop off on the ground, so they just be fall off. Okay, so they fall off and bust out, so falling from down from a height, which leads to a better dispersion of the seed. So if like some plants having a very large heights, like the 50-meter, 100 meters plants also here. So these 100-meter, 50-meter plant, when their fruit going to be burst on the floor, then they just burst out and the seed can be dispersed on the ground, and this will lead to the formation of new plants. There are also examples like the apple, camellia, canna, coconut, calabash, passion fruit. They are example of the plant which seed are dispersed by the gravity or force of attraction. Now this seed dispersal by explosion, that is the last seed dispersal mechanism we have to study. Explosion in fruit literally refer to bursting with all its energy. So when the fruit is got all ripened, so what happened with the fully fleshed ripened fruit? They will be burst out because now the internal mass that has to be very much broadly present inside. So the seed coat, no longer the fruit coat, no longer can be hold that. So at that time they just burst out. So this type of seed dispersal is mainly seen in those plants which having pods like okra, lupin, grouse, broom. There are a few examples of plants whose seed are dispersed by explosion. Pea, beans, and plants also have pods and which seed burst out when they ripen and pod has dried. So these can be the some condition for the explosion inside the fruit. So via this explosion, that seed just come out from their rooting body and dispersed in a place, and they will get to rise into a new plants. So this was all about what is the seed structure, how it is look like, and how they can be dispersed by several factors like insect, wind, gravity, water, explosion. So these all are the factors which going to help seed to disperse from one place to another place and increasing the diversity of fruits. Now, after this all study, I think you will be able to answer these questions. Like a few normal seedling of tomato were placed in a dark room. After a few days, they were found to have turned white color, like almonds. Which of these can be used to describe them? Okay. Gibberellin can facilitate seed germination due to their influence on an enzyme which can stimulate germination of barley seeds is during the germination of seed, the seed coat ruptures due to, I think this you can answer. The proteinaceous part of the maize endosperm is, just tell you that. One of these gases is required for the germination of pea seeds. Guesses which one of the gases will be required. Seed dormancy allow to plant to what? The protective covering over radicle during the germination of seed. I told you when we studied the maize plant. Which of these compound can induce seed dormancy? I think you know. An aluminum seed showing hypogel germination is so why. Still, if you wanted to have a more look on the seed structure, their dispersal mechanism, and how the seed grow, you can go through these references. They will definitely going to help you on literally a developed version of the seed, or you can say elaborated version of the seed structure and their functions. It's okay. That's all for today. I think you all got the understanding of the seed dispersal mechanism and the structure. Thank you.